All right, here we are going to talk about the neuron itself. The neuron is the technical name for the nerve cell, which is going to be our major communication cell in the nervous system. So, here's the thing about a neuron. While they do have some ability to regenerate depending upon the amount of damage they take, and if they're able to easily reconnect to the part that got cut off, they don't typically. As I mentioned in the last audio, they can, but it's not fast and it's not necessarily guaranteed. That being said, neurons themselves a individual cell lives a really long time. It can live and function for up to 100 years with proper care. And this is fascinating considering the fact that if we look at something like a red blood cell, its maximum lifespan is 120 days. So, one of the things that scientists are looking at is how are these nerve cells able to last so long? One of the things about them is that they are unable to divide. So to make new nerve cells, that is going to come from a stem cell. You will not be able to divide up the nerves in your brain to get new nerves. It has to come from a nerve stem cell. The other thing to note about neurons is that they have a very high metabolic rate, meaning that they are going to consume copious amounts of glucose and oxygen in order to do their daily minute functions. In order to do what they do, they need a lot of it. We'll talk about that in physiology, but one of the things that I show my students in phys is the fact that the brain specifically consumes approximately 50% of the glucose that we consume, that is used in our body. 50% of that is the brain, and it's something like 20 to 50% of the oxygen we breathe in goes to the brain as well. It is a very, very greedy organ. And it's not the glial cells that are going to be utilizing that glucose and oxygen. It is the nerve itself. Neurons can be sensory motor or mixed nerves. Sensory means that they are going to receive input. So if you put your hand on a hot stove, that heat, that pain, those are sensory neurons sending that information to your brain. If you smell a rose, you're listening to my voice right now, looking at your screen to see the slide that I'm on. Those are all sensory inputs. Those are all sensory nerves that are firing to bring that information to your brain. Motor nerves are going to take information from the brain and send it to what we typically refer to as an effector, meaning it's going to take it to a gland or an organ, a muscle, and tell it to do something. Release this hormone, contract, do something, an effector. So those are motor nerves. And then mixed nerves can either receive information or send information. So they can either receive input or they can send out output. As I mentioned at the very, very end of the last audio, the nerve is going to have a cell body, otherwise known as a soma. It is usually the largest mass of the nerve. It's typically going to have a spherical nucleus that kind of looks like an owl's eye with the nucleolus attached to it. If I were to dye it and view it under a microscope, it's going to be very granular or spotted in appearance, and that is going to be partially due to neurotransmitters. Most cell bodies are actually located in the spinal cord or in the brain. We do have a couple of collections of cell bodies outside of the central nervous system in the peripheral nervous system, and where those cell body clusters are typically referred to as ganglia. The clusters of cell bodies, when they're found in the central nervous system, are referred to as nuclei. So when you're reading those or you're hearing in physiology specifically, because we spend almost an entire modules talking about the nervous system in physiology. When you hear the term ganglia or nuclei, it's referring to the clusters of nerve cell bodies. Dendrites, remember, are going to receive information coming from either a receptor or from another neuron. They're going to receive that information and transmit it into the cell body. From the cell body, that information, that electrical signal, it's called an action potential actually, is going to be transmitted and generated down the axon hillock. The axon hillock is going to be a mound off of the cell body, which the axon is going to come off of. The axon itself is a very long, slender, almost looking like a fiber or thread that's going to be uniform in diameter across this entire length. Some axons are very, very short, especially the ones found in the brain. Some of them are very, very long, especially the ones found in the peripheral nervous system or in the spinal cord. But regardless of its length, it's going to be the same diameter the entire time. Now, from a terminology standpoint, the cytoplasm in the axon is actually referred to as axoplasm to tell you it's cytoplasm, but in the axon. And the cell membrane surrounding the axon is actually referred to as axolema. Do these come up frequently? From a technical standpoint, they do, but generally speaking, we just call it cytoplasm and cell membrane. At the end of the axon, we are going to have branches. These are typically referred to as terminal branches, or their technical name is telochondria. The ends of these branches are going to be bulbous, and these are going to have a variety of names depending on the textbook. Axon terminals, synaptic knobs, batons, which is French for knob, actually. And there's going to be many of these, many on each of the branches, and there will be many branches. What do these knobs do? These knobs are going to release neurotransmitters that are going to float from the neuron across a space called a synaptic cleft, and then send the signal onto something else. It could be another nerve, it could be whatever organ, gland, or muscle we want to do something, but it's going to carry the signal or transmit the signal from the nerve to the next stage. That is the purpose of these synaptic knobs and neurotransmitters. As I mentioned when we were talking about the glial cells, some of these axons are going to be surrounded by a myelin sheath. A myelin sheath is a fatty sheath, and it doesn't cover the entire length of the axon. It's broken up into little chunks. The spaces between these little sections of myelin sheath are referred to as nodes of Ranvier. And these nodes of Ranvier, along with the myelin sheath, allow for a fast, quick transmission of the signal down the axon. Which is why in autoimmune conditions, specifically like MS, or some viruses that affect the myelin sheath, attack the myelin sheath, it's problematic because we have these axons that require fast transmission, which they can do due to that fatty covering. They lose that fatty covering, they can no longer do their fast transmission. The transmission either gets lost or it's very, very delayed. So you might look at your hand and go, oh, I need to move my hand. And your hand moves 5 seconds later. Instead of, oh, I need to move my hand, and it moves immediately. That's if it even gets there at all. But that's only going to be a problem with axons that are supposed to have the myelin sheath, and not every axon has it. Axons that have the myelin sheath are typically referred to as white matter because that fatty covering gives it a very white appearance, and axons that don't have that myelin sheath are going to be referred to as gray matter because it looks gray. We did not get very inventive with our descriptions here, which is kind of nice. Because sometimes when we get inventive, It's very confusing. Now, that is your basic neuron. Those basic components can be configured in a couple of different manners. There is what is known as a unipolar neuron, and a unipolar neuron has no real true dendrite, but it is still going to have an axon, axon hillock, and still going to have our synaptic knobs down at the end. The traditional one that you've probably seen is going to be the bipolar. The bipolar neuron is going to have dendrites coming into the soma, but it's going to come in on something that looks like an axon. And it's still going to have its axon, and sorry, that was down here. It's still going to have its axon, and it's still going to have an axon hillock, and it's still going to have its synaptic knobs. Pseudo unipolars look very similar to unipolars, only the dendrites come in at the axon hillock as well as the axon leaving at the axon hillock. So that's what this is showing here. And then multipolar is the other one that you're probably you've seen quite frequently. It's got lots of different dendrites coming in at lots of different points on the cell body, but still only one axon with multiple synaptic knobs at the end. So, unipolars have a very short axon, seems to run right into dendrites, tends to be found in ganglia. These are typically going to be sensory in nature. Bipolar neurons, going to have a distinct dendrite and separate axon. These are typically going to be found in special sense organs where they act as receptors. What are special sense organs? Not something that we really cover in this particular section, but this is going to be your eyes, your ears, your nose, your sense of smell, your sense of taste, your vision, your hearing. That's bipolars, bipolar neurons, I should say. Multipolars, multiple genetic processes from one axon, typical and most common. Find them all over the peripheral nervous system, all over the brain, all over the spinal cord. They're everywhere. All right. So the last thing I want to talk about the structure of a nerve is a neurotransmitter. What is a neurotransmitter? A neurotransmitter is a chemical messenger that is released from the synaptic knobs of a neuron that crosses a synaptic cleft to a receptor that is found on another nerve or on a effector, organ, gland, or muscle. The most common neurotransmitter is acetylcholine. Acetylcholine is critical for muscle contractions, but it's also used in a couple of other places. Another extremely common neurotransmitter, especially for the central nervous system, is going to be norepinephrine. However, other neurotransmitters are going to be things like dopamine, serotonin, histamine. I know histamine gets a bad rap because of allergies, but it is also a neurotransmitter. GABA. There's tons of neurotransmitters. Some of them play very specific roles, so you only find them in very specific places in the nervous system, and others we find they have widespread roles, like acetylcholine and norepinephrine, and we find them in many, many places. Depending upon the neurotransmitter, the receptor that they bind to will have a very specific name. For instance, norepinephrine. The receptors for norepinephrine are going to be referred to as adrenergic receptors. This is something we will talk about more in physiology, but I do want to give you a heads up here. There are half a dozen or so different type of adrenergic receptors. There's alpha 1s, alpha 2s, beta 1s, beta 2 adrenergic receptors. There's a couple other ones that aren't as common. And you're going to hear about them because a lot of medications affect the adrenergic receptors, alpha 1s, 2s, beta 1s, 2s, etc. Acetylcholine, you're also going to hear about them because we have many medications that affect acetylcholine receptors. Depending upon if it's a fast or slow receptor depends on its name, but one that you'll typically hear is a nicotinic receptor. Not nicotine, nicotinic. And a nicotinic receptor is a receptor that accepts acetylcholine. And obviously, there are other ones in here. These all have their own specially named receptors to tell you that this receptor goes with this neurotransmitter. So just be aware of it. That is actually going to finish off this particular audio. The next thing that we are going to do is we are going to start talking about the spinal cord.